Let's begin our video on section 4.4 on resting membrane potential. Here are your learning objectives, and we are going to start with defining what is a resting membrane potential. What are we talking about here? A plasma membrane is going to establish and maintain something called an electrochemical gradient, or its resting membrane potential. So when I say that term electrochemical gradient, we're talking about a gradient of electrochemical potential. Now, there's two parts that makes this up. We have the chemical gradient, which is referring to the difference in solute concentration across a membrane. So for instance, if you're looking at the illustration down here, we're looking at the, let's say, potassium gradient. So we have lots of potassium ions inside our cell here and not so much outside of our cell. That's what we talk about in a chemical gradient. And the other part is an electrical gradient, which is the difference in charge across a membrane. So notice how we have these plus signs here outside of the cell and negative signs inside the cell. That means the inside of our cell is more negative compared to the outside of our cell. And that helps create our electrochemical gradient. This potential is essential for our muscle and nerve functioning. So let's do an introduction first. Our membrane potential is going to be the potential energy of charge difference. And when our cell is at rest, we call this a resting membrane potential. It's the potential when the cell is at rest. Simple as that. And we have two conditions for this resting membrane potential. First, we have an unequal distribution of ions and molecules across the plasma membrane. So just like we saw in that previous photo, we are going to have more potassium inside the cell in the cytoplasm than compared to the interstitial fluid or extracellularly. And extracellularly, we're going to have more potassium. More sodium. And you could see this in the illustration down here showing us our ion concentration gradients, that we have more sodium outside of the cell than compared to inside of the cell. And this is all due to the sodium-potassium pump, which you don't see pictured here. But if you remember from our previous section, we will have 3 sodiums pumped out of the cell and 2 potassiums pumped in. So it helps to maintain these ion concentration gradients. And the second condition for our resting membrane potential is the unequal relative amounts of positive and negative charges. We'll see that we have more positive charges on the outside than on the inside of the cell. And the reason for that is that our sodium ions are so numerous here and have a positive charge. And on the inside of the cell, even though our potassium ions have a positive charge, they really don't make that large of a difference compared to these really large negative proteins that it is attracted to. So the inside of the cell is actually negative because of these really large negative proteins. So if you haven't gathered from the previous slide, the most important ions here are sodium and potassium. Their movement depends on that electrochemical gradient that we discussed. So first we'll mention our role of potassium. This is the most important determinant in the specific value of our resting membrane potential. Potassium is going to move down its steep concentration gradient through leakage channels from the cytosol to the interstitial fluid. Our negatively charged proteins are going to stay inside our cell though. And the electrochemical gradient is that our positive charge outside is going to repel the movement of these potassium ions out of the cell. So it is going to want to stay in here. The negative charge on the inside attracts the potassium ions inside. And what we'll see is that the potassium ion will move until an equilibrium is reached. So while we do have some potassium ions that'll move outside of the cell, the majority of it will stay inside the cell. There's another one. And another one, I guess. Now let's talk about the role of sodium. Sodium is going to diffuse into the cells from the interstitial fluid to the cytosol, simultaneously to the loss of potassium. So we don't show potassium moving out of the cell here, but that's what would happen. And sodium is moving through sodium leakage channels. So that's what's represented by this rectangle over here, and it's moving down its concentration gradient. We have a high amount or concentration of sodium outside the cell, and it wants to move to an area of low concentration, and it's pulled by the electrical gradient, right? Sodium is a positive ion, and it is attracted to the negativeness inside the cell. Our leakage channels prevent as much sodium into the neuron that a potassium would move out. So the inside is going to become more positive as sodium moves in. So when we talk about a neuron, we'll eventually talk about how an action potential or a nerve impulse takes place. And essentially what happens is a lot of sodium move through these channels inside the neuron cell to make the inside very positive. And that's what triggers a nerve impulse. Now, how do we maintain a resting membrane potential? We mentioned how our sodium-potassium pump is so significant in doing this. It helps to maintain that potassium and sodium gradient following their diffusion. We are going to have our potassium pumped out of the cell, and our potassium attaches and gets pumped into the cell. And so notice they are in opposite directions and against their concentration gradient, and ATP is necessary because this is an active process, an active transport.